Good afternoon. My name is Jennifer Sorrell, and I'm the Executive Director of the UCI Anti-Cancer Challenge. Thank you all so much for joining us for today's Cancer Research Update. If you'd like to ask a question during today's session, please use the Q&A feature at the bottom of the screen. Next slide. Now I'd like to introduce the Director of the UCI Chow Family Comprehensive Cancer Center, Dr. Rick Van Etten. Dr. Van Etten also serves as a professor of medicine and biological chemistry in the UCI School of Medicine and as a senior associate dean and associate vice chancellor in the UCI Susan and Henry Samueli College of Health Sciences. As director of the only Orange County-based comprehensive cancer center designated by the National Cancer Institute, he leads a team of more than 200 physicians, scientists, and other health science professionals, all dedicated to defeating cancer. He is a fellowship-trained hematologist and leukemia researcher who is regularly featured as a physician of excellence in Orange Coast Magazine and as a Castle Connolly top doctor and whose own blood cancer research has changed lives. You may also know Dr. Van Etten as a cyclist and founder of the UCI Anti-Cancer Challenge, a ride, run, and walk to raise funds for promising cancer research. I'll now turn it over to Dr. Van Etten.
Well, thank you very much, Jennifer. I'd also like to thank everyone who's attending today to this research update for the Anti-Cancer Challenge, which will help showcase some of the amazing research that has come out of the money raised by participants in the Anti-Cancer Challenge last year. If I could have the next slide. I just want to remind folks who we are at the UCI Chow Family Comprehensive Cancer Center. We're an NCI designated comprehensive cancer center, the only one based in Orange County. And in that role, we serve as a vital resource for Orange County and all the surrounding areas in the fight to alleviate the burden of cancer. We integrate world-class research, prevention, and the most advanced diagnostics, treatment, and rehabilitation to provide the best possible care for patients and families. We provide the largest number of clinical trials in Orange County, which is the only way for patients to get access to the very latest drugs and devices before they're approved by the FDA. And I'm proud to announce that last year, our National Cancer Institute designation was renewed competitively for another 5-year period. Next slide. We have a couple of new programs at UCI Health and our cancer center that we're very proud of. One is a hematopoietic stem cell transplant and cell therapy program, which was launched now just about 3 years ago. It's Orange County's only adult bone marrow transplant program, so patients no longer have to travel long distances to get this potentially life-saving therapy. We have ongoing cell therapy treatments with so-called CAR T cells and other immune therapies. We were accredited by FACT, the national organization, last year, and we recently obtained CalOptima Center of Excellence recognition. I'm just showing here a picture of Dr. Stefan Sharia, who's our program leader. Next. Another thing that we're very excited about, which has emerged from the research of our members, is a new imaging technology. You can think of it as a super CT scan, which can image the human body at 100-fold resolution with a greatly reduced X-ray dose. This really changes the paradigm for screening for cancer, including cancers that we haven't previously had the ability to screen for, one of which is pancreatic cancer, which is going to be talked about by our two researchers today, and also has applications in cancer radiation therapy. So we're really excited about this new technology. Next slide. One of our missions as an NCI Cancer Center is to alleviate the burden of cancer on Orange County, which is our catchment area. These maps of Orange County, these heat maps of Orange County that you see on the left are the rates of screening for 2 different types of cancer, cervical cancer on top in the green, and colorectal cancer on the bottom in the blue. And the light areas are where the screening rates fall far below what the US government and the National Institutes of Health are recommending as our goals. So to address this, next slide, we have put together a program which we call Advancing Cancer Care Together. And it's based on UCI Medical Center up in Orange, partnering with 3 federally qualified health centers in Orange County. These are centers funded by the federal government to help serve underinsured and uninsured patients. And it is the brainchild of Dr. Rob Bristow, who's shown here, and also Dr. Sora Tanjasiri. And it's a hub and spoke model where we bring state-of-the-art cancer screening and prevention out to these federally qualified health centers for the first time. And this program has already resulted in its first 2 years in a dramatic increase in screening rates for these cancers in these community health centers. Next slide. We're also very excited that UCI Health and the Cancer Center are expanding their footprint within Orange County. We have a new cancer center, which is under construction right now at the corner of Jamboree and Campus Drive down in Irvine. This is just a picture of the construction site. If you click forward, our new cancer center, this is what it's going to look like, will open in the summer of 2024. It will be immediately Orange County's most advanced cancer treatment center. It will offer medical oncology, radiation therapy, cancer clinics, imaging, pharmacy, infusion center, and also, importantly, access to clinical trials and clinical research. Next slide. So why the Anti-Cancer Challenge? It's a mechanism that I launched as director, which provides much needed discretionary funding for me to be able to fund the most innovative, high risk, high payoff research that comes out of the laboratories of our members and our scientists. So participants sign up, and it's based around an athletic challenge with a 5-10K run walk and a multiple cycling events. All the funds raised go to support promising research at our cancer center and our pediatric cancer partner, Chalk Children's. We have corporate partners that generously underwrite the event costs to allow us to fulfill our pledge of passing all the money on to research. The event has had over 11,000 participants since its launch. We've raised over $3.7 million for cancer research, and the event has consistently been ranked in Orange County Business Journal's top fundraising events. Next slide. So how do we use the money? We have it really in two different ways. One is pilot projects, which are 1-year grants to our basic and translational scientists to provide funding to allow them to generate the critical preliminary data that they need to compete successfully for an extramural grant, for example, from the NCI or from the American Cancer Society. Next slide. The other example, track 2, is to support early phase clinical research. I should have mentioned that track 1 research, you'll see an example of that when Dr. Halbrook talks in a minute. Track 2 is to support early phase clinical research. This is the very first time that the discoveries of our scientists are moved forward into the clinic where they can be impacted and be tested in humans for the first time. And this slide just shows the various stages in the development of the clinical trial. These funds are basically focused on what's called phase 1 and a little bit of phase 2. And you will hear from Dr. Maheswari Senthil an example of how these funds are used for this purpose. Next slide. So the program has had tremendous impact since its inception in 2017. As I mentioned, $3.7 million in research grants awarded. That has had a dramatic return on investment. So you can see that through this funding, over $33 million in new extramural grant funding to our cancer center has been awarded. Furthermore, there have been 2 startup companies that were launched with Anti-Cancer Challenge funds, 5 clinical studies that have addressed specific community needs, and 6 additional clinical trials that are accruing patients currently. So we're very proud of this track record, and you can see some of the vignettes of funded research on the right-hand side. Next slide. So without further ado, I would like to get into the main program, and I'm very pleased to introduce Dr. Chris Halbrook, who's an assistant professor in the Department of Molecular Biology and Biochemistry in the UCI School of Biological Sciences. And he's going to talk a little bit about the work supported by the Anti-Cancer Challenge on pancreatic cancer. Dr. Halbrook.
Hello, I'm really excited for this opportunity from the Anti-Cancer Challenge to be able to share some of our research in my lab with you, and Dr. Van Etten for allowing me the opportunity to do so. I just wanted to give you guys a quick background of why I'm here. I'm a new assistant professor at UCI. So I've worked in pancreatic cancer ever since I started my graduate training at the Stony Brook University in the Mayo Clinic. And this was about 10 years ago. You can see me in the background here back when I was still full of youthful vibrance and a little bit more hair. And then I transitioned to the University of Michigan to join what has really become one of the largest pancreatic cancer research teams in the country. where we were really pushing the envelope on a daily basis in terms of how we're discovering cures eventually for this disease. But when I was looking for the place where I would go start my own independent lab, I realized that UCI was a very set up to be a very good place to do groundbreaking research, but we didn't have anyone that was focusing on this disease at the time. So this, I think, is going to be the next, hopefully, big site that we'll see that the West Coast will have its own contribution to where some of the other larger groups like the University of Michigan, MD Anderson, and the groups in Boston have really been the center of leading the field in terms of this particular cancer. So why am I focused on pancreatic cancer? So this is a list of different cancers that people are expected to be diagnosed with, projected up until 2040. So pancreatic cancer is actually way down here. So you may think to yourself, this isn't that big of a priority. But if you look at the number of people who are actually dying from cancer, you can see that pancreatic cancer is sitting here right now. So we're about here. We're currently the third leading cause of cancer-related death. And this is projected to become the second leading cause of cancer-related death by about 2030. So we're getting less people are dying of other cancers you're more likely to be diagnosed with because we're getting so much better at treating it. But when it comes to pancreatic cancer, we're getting around the same number of people with a progressive increase, but not exponentially increased. But everyone who gets the disease so far is not faring very well. So again, I've been in this particular field now for about 12 years, and this is the first presentation I ever gave. I went and pulled up the introductory slide for this. And at that time, the 5-year survival rate for pancreatic cancer was 5.5%. We didn't really know the major causes of the disease other than smoking and chronic pancreatitis. And at the other end of it, there's which is now, I think we realize about 10 to 20% can have familial pancreatic cancer disease causes or underlying genetic predispositions to it. But that means about 80% of people who get pancreatic cancer, we don't have any idea why they're getting it. And when we're talking about pancreatic cancer, I'm talking about a disease called pancreatic ductal adenocarcinoma. So this is where we moved to in the 12 years I've been in the field. There was a big goal where there wasn't originally a lot of funding for this disease because it was considered essentially a death sentence. So why would Congress want to put money into this? So in the early 2010s, there was a passage of an act called the Pancreatic Cancer Act, basically saying that if we have diseases that are this deadly, why are we not putting more money into them? And so there was a goal over the 2020s that we were going to double pancreatic cancer survival from this 5% figure by the 2020. And so we've been able to achieve that, and we're still progressing up slightly per year, but obviously we're still failing the vast majority of patients. But this level of the disease that was originally 5.5% had stayed at that for the previous 30 years. So we're really finally starting to make some progress in treating this disease. So I had a really unique opportunity recently where I was approached by one of the flagship journals that publishes biomedical research called Cell. And they asked me and several colleagues of mine from the University of Michigan and one of the pioneers of the field from the MD Anderson Cancer Center, what we really think have been the biggest things that have changed in our understanding of pancreatic cancer. And so in this time frame, we've really seen an idea of knowing exactly what the mutations are that drive this disease, how the cells in the pancreas, which cells are the ones that are actually mutated and will lead to the disease, the precursor lesions that we could hope to eventually screen for, and then billions of dollars that was spent sequencing tumors in order to figure out that there was various subtypes of tumors, which unfortunately so far have not actually really informed how we can treat different patients. The other thing that has become abundantly clear as we continue to research this disease, so this is what we call a hematoxylin and eosin section. So this is a slice of a tumor that we stain with a couple different dyes to try and just see the architecture of a tumor. And what I'm bringing this to your attention for is this thing right here, this little island of these particular looking cells. These are the cancer cells in a pancreatic tumor. The rest of the stuff that's not these little islands is actually stuff that is not cancer cells. So there's other cell types in there called fibroblasts, there's immune cells, and there's a lot of this is just filled up with extracellular matrix. So things like collagen that make the tissue really stiff. And it's one of the things that a pancreatic surgeon can tell you when they take one of these tumors out, is that this fibrosis of the extracellular matrix is essentially scar tissue. And these tumors are eventually as hard as a baseball when they get taken out. There's so much of this scar tissue that's in the tumors. So what we've actually, and others, is really a big focus of the field, have done is looked at if the cancer cells aren't the major cell that you actually find in the tumor, what are the other cells in the tumor doing to actually shape how these cancer cells are getting their signals to grow? And so there's been, again, just an absolute mind-blowing amount of heterogeneity that we found. So different cell types within individual cells that we found. So within the things that I was introducing that are fibroblasts, we found that fibroblasts are not just fibroblasts. There's actually at least 3+ different types of fibroblasts you can find in each region of a tumor. These are different between patients. There's different types of immune cells that exist within tumors. And each one of these lines indicate different interactions that different studies have shown that can be targeted therapeutically to potentially take apart this crosstalk network between the different cell types that support the cancer growth in the tumor. And the other thing that's become abundantly clear with these lines this way is showing you that these are interactions that are actually inhibiting the immune cells that you'd like to see in a tumor. So these CD8 T cells, these are the cells that would respond to immune therapy. You do not find these in pancreatic tumors, and these are one of the tumor types that just do not respond to immune therapy as much as you've seen this be a miracle drug in some other tumors like melanoma on occasion. So again, the amount of different heterogeneity between the cells, so the different cells within an individual type has been just one of the biggest things that the modern technology that we're allowed to use in research versus the technology we had 10 years ago is really uncovering. We can see different cell types that are in a normal pancreas that expand into different cell populations in the tumor. And we found that some of these are actually beneficial for the tumor cells, so we want to get rid of those. But some of them actually have roles in preventing the cancer from metastasizing, so we don't want to get rid of those cells. And there was classic clinical trials for pancreatic cancer in the early 2010s where we didn't know there was this many different types of these fibroblasts that exist within a tumor. And so targeting all of them actually let the cancer metastasize faster, which in a tumor that kills as fast as this does, it was really a sad result for a clinical trial. So we've really come up with an idea of what we consider the way that we're going to eventually cure this disease. So we know the mutations that drive the disease because we've sequenced it. There's finally over 30 years, people have been trying to target this particular protein. And in the past 5 years, we've seen compounds finally be developed. In the past 3 years, they've been deployed in the clinic. And now we're finally seeing the data that is coming from patients being treated. It looks fantastic. We know that there are patients that respond to other therapies because they have these familial drivers, which again, years ago, we weren't unaware of. There's specific features of pancreatic cancer cells that we can target. All the other cells in that tumor microenvironment, we call the surrounding area around the cancer cells that support the tumor growth, how we can leverage the interactions that are in there to block the ability of them to suppress the immune response that we want, so we can actually use immune therapy in these tumors. And eventually we're going to translate these into innovative clinical trials so we can get patients into trials that work for them faster as opposed to just having them sit on trials that aren't working for them. So I focus more on basic research in which my lab focuses on the mechanisms by which these parts of how we're going to eventually cure pancreatic cancer are focused and how these different things will work together. So over the past 7 or so years, we've actually focused a lot on not just how cancer cells can communicate in terms of signals they exchange, but also in terms of the way that they share nutrients. And we found that this has incredibly profound effects on different aspects of the tumor biology. So those fibroblasts I was talking about, these are cancer-associated fibroblasts, can feed pancreatic cancer cells certain nutrients. We see macrophages, which can be about 40% of all the cells in a tumor, can directly block the therapy that we use for chemotherapy. We've shown mechanisms by which these cells get programmed. We can see the cancer cells can actually eat that matrix that I showed you. We can see there's different types of cancer cells that act to support each other. And eventually, we've also shown now that these macrophages are directly blocking the ability of these T cells you want to come into the tumor by degrading the nutrient that they need. However, almost all of the work that we've done so far is focused on what would happen in a pancreas tumor. But when most people are diagnosed with this disease here, they don't have this localized disease. Only 12% do. If you have localized disease, your 5-year survival rate is much better than the people who get diagnosed with metastatic disease here, which is about over half of the patients. These are the patients who do the absolute worst. And so these are the patients that we actually need to focus on treating. So what we've done through the money that we've obtained through the Anti-Cancer Challenge is we've used mouse models that we've developed for pancreatic cancer to ask the question is what is different with a tumor that is in the pancreas to what happens when it metastasizes. And the liver is the most common place you see a tumor metastasis in pancreatic cancer. And so this is working with a good collaborator at the Henry Ford Pancreatic Cancer Center. This work's done by a very talented graduate student in my lab. So we're using a technology called single-cell sequencing, which allows us to look at all the genes and how they change on an individual per cell basis. This is one of those things that has really just revolutionized how we think about different cells in a tumor in the past 5 or so years. So these data are very complicated. So each one of these dots is a cell that we have the entire expression level of every single gene on. So this becomes a big problem of trying to figure out computationally what to do with this much data. But we've developed ways to be able to identify what each of these individual cell types are. This is a visualization that as they sit closer together, they have shared features, which we can eventually then say that, for example, these red cells that you find in both the pancreas and liver tumors are those fibroblasts that I was talking about. We can see the different proportions of the cells that exist within these tumors. We can actually look at within the cancer cell populations themselves. We can see each one of these vertical lines is a cell, each one of these horizontal lines is a gene. When you see these blocks of yellow, these are shared features that we're allowed to say that these are able to say that these are different populations of cancer cells, which have different features that we can target with different drug types. And one thing that was very important for us to find with this is you can see this green bar in the liver tumors accounts for over half of the cancer cells versus the pancreas tumors. And the ones that are these green ones here have very specific pathways that we think we can target with different compounds that we've accumulated to do this with. And the last population I wanted to highlight that was very interesting for us. So there's this big focus on these cancer-associated fibroblasts that are in the primary tumors. But what we found so far when we're looking at our liver tumors in comparison is that there's these pink cells right here that almost entirely are located in the liver tumors here. And if you look at this distribution, almost half of them in the liver tumors don't fit any of the things that have been described with every thing that people have looked at with the primary pancreatic tumors. So essentially the biology of the liver tumor is completely different than that whole network of interactions that I've been showing you before and is still relatively undiscovered. So we've shown these fibroblasts are real. I won't have time to get into it. We're working with a really talented investigator in the developmental cell biology department here who's developed these algorithms to predict the interactions between cells. And we can see our cells of interest, how they are talking to other cells, how cells are talking back to them, different networks that we can use to target this. And again, this is done in collaboration with the postdoc in Max's lab, Ying Zi. So again, this is still early days with this, but that's the whole purpose of this mechanism is we've generated enough data now that we're putting in grants for long-term studies involving this whole thing. We've also seen so much that there is left to discover in this, and this is only possible through funding through the Anti-Cancer Challenge. So with that, I just want to quickly wrap up to thank the people who are in my lab right now. Again, this work was largely done by a PhD student, Rima Singh. We've been very fortunate to have our work funded through several mechanisms. Again, most important for this project is the Anti-Cancer Challenge. And then we also have our lab and myself Twitter accounts that you can look to actually see different things that are coming out of the lab on a regular basis. So I encourage you to also follow us there. But with that, I'm happy to also answer any questions that you have on this and other things to do with pancreatic cancer as well when we have our Q&A session. So with that, I will cede control back to our organizers here, and again, thank them for this opportunity to talk to you.
Thank you, Dr. Halbrook. If you do have questions for Dr. Halbrook, please use the Q&A feature at the bottom of the screen, and he'll answer them after our next presentation. So I'd now like to introduce our second speaker, Dr. Senthil is the Chief of Surgical Oncology and the Director of Peritoneal Malignancy Program at UCI. She is the Medical Director of Center for Clinical Research at UCI and John Wayne Cancer Foundation's Endowed Chair. Dr. Senthil is a surgical oncologist with special expertise in the management of patients with peritoneal carcinomatosis and advanced malignancies. The overarching goal of her practice is to improve the outcomes of patients through innovation, research, and technical excellence. Dr. Senthil is a clinician scientist, and her research is focused on changing the treatment and diagnostic paradigms of cancer. She is working with a team of outstanding scientists at UCI and other institutions to develop an in vitro 3D vascularized microtumor of peritoneal carcinomatosis to assess treatment response. She is also developing a liquid biopsy utilizing exosomal biomarkers in patients with gastrointestinal cancers. Dr. Senthil is the principal investigator of the Stopgap Phase 2 clinical trial that is evaluating a new strategy with intraperitoneal chemotherapy to treat patients with gastric peritoneal carcinomatosis. Dr. Senthil is a highly respected educator. She founded the Complex General Surgical Oncology Fellowship, both at Loma Linda University Health in 2015 and UCI in 2021. She has received numerous awards for her commitment and dedication to educational excellence, including a Golden Apple Award, Arnold P. Gold Award for Humanism and Leadership, and John Connolly Teacher of the Year Award. She is also a recipient of Alpha Omega Award. Dr. Senthil mentors several students, residents, and fellows on the clinical and research fronts. She's also published over 100 peer-reviewed papers, several book chapters, and serves as the editorial board member of Surgical Oncology. I'll now turn it over to Dr. Senthil.
Thanks, Jennifer, for that kind introduction. Thank you all for participating today. First of all, I want to start off by truly expressing my heartfelt gratitude for not only your presence here, but your continued support to the Anti-Cancer Challenge. As Dr. Rick Van Eden mentioned, your support is what drives a lot of this yearly clinical trials before it becomes funded project. You can see the success of this kind of philanthropy does to research. And I'm extremely grateful to have received 2 of this, and both of the projects I'm going to give an update about today. Once again, your continued support to the Anti-Cancer Challenge really promotes research that is life-saving and also helps investigators like me to get the initial data to move it to the next phase. So thanks again. Okay, I'm going to... Now, you heard this long terminology that Jennifer kept on mentioning during my introduction. It's called as peritoneal carcinomatosis. I'm sorry if some people are sensitive to the picture that's on this slide, but I wanted to put that there to show the extent of these tumors. So peritoneal carcinomatosis is a metastatic manifestation, which means that the cancer that originated in an organ has moved out of the organ to diffusely spread in the peritoneal cavity, which is essentially the abdominal cavity. So this is an abdominal cavity of a patient who had peritoneal carcinomatosis because I removed it. So you can see here, if you could see the mouse that I'm moving, that's all tumor. The whole thing is actually tumor. And then this patient had multiple other areas of tumor as well. So it's not like a specific organ that it goes to. It's not like a liver metastasis or a lung metastasis. This is very diffuse metastatic process. Now, unfortunately, this particular type of metastasis is the deadliest metastasis in gastrointestinal cancers. Even Dr. Halbrook mentioned about pancreatic metastasis. Pancreatic cancer can do this type of metastasis as well, and it is extremely deadly type of cancer spread. But what I focus on is this type of cancer spread in GI cancer, specifically colon and gastric cancer. The incidence of this kind of cancer spread, like peritoneal carcinomatosis in colorectal cancer, is about 25 to 40%. And in gastric cancer is even higher. It's about 30 to 50%. So 30 to 50% of patients who are diagnosed with gastric cancer will end up with this devastating metastatic process, which has an extremely short survival times, and I will show you that. And these are inherently resistant to treatment, this particular cancer. Now, since the clinical trial that I got funded through Anti-Cancer Challenge is for gastric cancer, I just want to further elucidate the background of gastric carcinomatosis. So incidence of gastric cancer spreading to the lining of the abdomen is about 30 to 50%. This is one of the patients who is enrolled in STOPGAP trial. So that's a clinical trial. So this is an laparoscopy image of this patient's abdomen. And what you're seeing here, this is the liver of the patient, which is the purple organ that's located here. And all this white patchy areas that you're seeing, a plaque-like distribution, is gastric cancer that has spread to the lining of the abdomen. Now, in this situation, even with best systemic chemotherapy treatment, even in the latest publication in Lancet, the survival is only about 10 to 12 months. So majority of the patients will not see the next calendar year, which is really strikingly sad. And systemic chemotherapy alone is not sufficient, and surgery is typically not considered due to the overall poor outcomes in these patients. Now, in Orange County, we have a large population of Asians as well as Hispanics who definitely have a higher incidence of gastric cancer and also a higher incidence of peritoneal carcinomatosis. So the purpose of the clinical trial that I put together is essentially to address this community need that we have with a large population of patients with high incidence of this particular cancer. And then the lack of adequate treatment options for these patients and making sure that we are able to enroll them in a clinical trial that would allow us to treat them better, right? So if chemotherapy that is given IV alone is not sufficient, the question then comes, how can we improve their outcomes? So this clinical trial actually has three components. And this is the trial protocol that was recently published in BMC Cancer, so that it is readily available to any investigator who would like to replicate this protocol. So what this treatment includes is that patients, after receiving 3 to 4 months of IV chemotherapy, will then get what is called as an intraperitoneal port. So if you look at this cartoon, there is a peritoneal access port. So essentially like an IV port, it is a portal with a catheter that is going into the abdominal cavity. And you can see the image of the portocath as to what it looks like. So it has a chamber, it has a catheter, and the catheter is in the patient's abdominal cavity. And we give both IV chemotherapy and intraperitoneal chemotherapy for patients enrolled in this study. So they get the standard chemotherapy, the typical chemotherapy that is effective in the setting of gastric cancer. And then there is also chemotherapy delivered directly into the intraperitoneal space. They get this treatment for about 4 cycles. So it's approximately 3 months of treatment. And then subsequently, we will reevaluate. So this is the trial protocol. So essentially shows that patients with gastric carcinomatosis are going to end up getting that IV chemotherapy after that initial laparoscopy and port placement. But more importantly, after the 3 months of that IV intraperitoneal chemotherapy, we do another laparoscopy and look for patients in whom surgical resection would be reasonable. Now, as I had mentioned previously, surgical resection is not indicated in the setting of gastric carcinomatosis in the current standard of care treatment paradigm. However, in a clinical trial, we could potentially offer surgical treatment for highly selected patients because there is evidence, not level 1, but however, level 2 evidence suggesting that there could be improvement in survival in patients who are appropriately treated and have entire cancer removed. So we do what is called as a cytoreductive surgery and heated intraperitoneal chemotherapy in selected subset of patients in whom the cancer burden is low and that the cancer can be completely removed. And so thus far, we have enrolled about 14 patients in this clinical trial, and nearly 40% of the patients have had good response to the point that I'm able to do this kind of surgery for them. And then all the patients have been able to complete treatment. So we have been able to establish the safety and feasibility of this approach in this very, very challenging disease process. And hence, I am now putting together the next large randomized control trial. So the data from the trial that was supported by the Anti-Cancer Challenge has now led on to the next large randomized control trial, which is going to be the important evidence we need to make practice recommendations, right? Because practice recommendations don't change unless we really do a randomized control trial in which current treatment option is compared to an experimental treatment option. And that's what we are proposing, and I'm hoping that that would be started very, very soon. But that's about the clinical trial that we're doing, and it's been life-changing for many patients who have been enrolled in this clinical trial. The next part, this is also another study that was funded by the Anti-Cancer Challenge. It's more of the basic science project. Now, as I had mentioned about peritoneal carcinomatosis, it's an extremely challenging disease, and it is inherently resistant to treatment. So we have to figure out ways to improve the treatment approaches and understand what treatment actually works in this particular disease process. Now, in order to test the effectiveness of the drugs in these tumors, we need proper treatment models. And you saw Dr. Halbrook's presentation where they are using animal models to figure out as to how we can understand, one, the behavior as well as responses, right? Now, typically animal models are expensive, and there are, mice and rat are not humans. There are also changes, and there's limited translatability to humans. So we certainly are looking for alternate ways to develop tumor models that replicate the human tumor microenvironment. And as you know, FDA recently announced in 2022 the 3R project, which is reduce, refine, and replace, or reduce, replace, and refine animal models, right? So we are definitely trying to reduce the use of animal models to test treatment responses and figuring out, in vitro tumor models that are closely resembling the human tumor microenvironment. So that has been a focus of some of my other work. And this is in partnership with Dr. Chris Hughes at UCI. So we have developed what is called as a vascularized microtumor specifically for peritoneal carcinomatosis. There are other in vitro tumor models, but there are significant advantages in the model that we are working with, which is the microtumor model. So this model actually is a three-dimensional model. This is a rendering of what this model looks like. It is a micro model, so it actually fits in a 96-well plate. But this model has stromal cells, tumor cells, endothelial cells, and also a living vascular network. So essentially what you're seeing here is in green is the blood vessels, and the red are tumor nodules, essentially in this case. And the tumor is growing within the vascular network of this model. And there is also stromal cells, like the importance of non-cancer cells in tumor microenvironment was also highlighted by Dr. Halbrook. So we are using stromal cells also in this, and we can use the lymphocytes or the white blood cells also in this tumor. Now, this is the model that I showed you is the original model, but we wanted to change this model to replicate the peritoneal cavity. So peritoneal cavity, unlike the usual tumors, it actually is a very confined space. So essentially, so what you're seeing here is the cavity, and then this is the peritoneal lining, and there is blood vessels are on the bottom of this area. So what we are showing is actually a redesigned peritoneal dual chamber model, where this blue area is actually the peritoneal cavity, and the red area is the blood vessels below that peritoneal cavity. In the second image, what you're seeing is the colon cancer peritoneal carcinomatosis tumors growing in this model, as well as the blood vessels actually penetrating into that mesothelial layer, which again shows an activity, a tumor growth activity here. And what we have used this is to create this kind of a test. So we are using this vascularized microtumor model to assess in vitro drug response and correlate that with the patient. So we will take a tumor from the patient, create this vascularized microtumor model. The patient will continue to get a standard of care treatment, but in the lab, we will be testing various drug and drug combinations on the vascularized microtumor, figure out what is working best for that particular tumor, and then be able to correlate that with the patient's response and say, is this model close enough for us to figure out if we can use this model to inform treatment decisions for patients. So this is in the development. This study is now approved, and it is actually listed in clinicaltrials.gov. In fact, my first patient has been enrolled today to participate in this clinical study as well. So that's all I have, and I'll be happy to take any questions.
Thank you so much, Dr. Senthil. So we do have a few questions here, and I encourage folks to continue to enter them into the Q&A on the bottom of your screen. So first, this question is for Dr. Halbrook. Do you have any clinical trials at this time that patients can participate in? And then specifically, are you enlisting people in your pilot project for pancreatic immunotherapy treatment? And then how does one enlist? And does this require you not to be on standardized chemo treatment?
So again, I'm a basic researcher, so I don't treat patients, but we do have a very active clinical trial program here with pancreatic cancer, and our whole hepatobiliary program has a big group of things. And again, I'm not the person to talk to on this. So Dr. Jennifer Valerin is one of our primary oncologists that handles this. And again, she's running tons of clinical trials that are going to be available for people that are available for standard therapy, or people who have failed standard therapy are not allowed on to those anymore. And there are several that involve forms of immune therapy that are on there as well. But again, I'm not the person who can give the best advice on things when it comes to treatment.
Sure, thank you. I wonder, Dr. Senthil, do you have anything to add to that question?
As Dr. Halbrook mentioned, we do have a very large portfolio of treatment for patients with any form of GI cancer and for pancreas cancer. There are a lot of even tumor agnostic clinical trials that engage in using immunotherapy in cancers, regardless as to what the tumor is arising from, but essentially, looking at some of the molecular features. And Dr. Jennifer Valerin, who's our pancreatic cancer expert, would be a great resource in terms of specifically addressing the question about whether current standardized chemotherapy, if a patient is on current standardized chemotherapy, would that be an exclusion criteria for the patient to be participating in a clinical trial? Those are things that we could definitely look into the eligibility criteria for anyone who's interested in participating.
Yeah, and I also forgot to mention, Dr. Farsid Dayani is also running a lot of clinical trials that enroll pancreatic cancer patients as well. So there's several oncologists here that have a participation in this group that is specifically trying to enroll pancreatic cancer patients on.
And I do see a couple of questions here. Jennifer, would you like me to answer them?
Sure. So this next one is for you. So why are some populations more at risk for stomach cancer?
Yeah, that's a great question. Great question. So the risk of stomach cancer has many factors, right? So in Hispanic people, we believe exposure to H. pylori might be a factor that might result in increased risk. There is documentation that there is increased incidence of this very specific infection called as Helicobacter pylori in our immigrant population. Definitely, that is that infection predisposes or increases the risk of gastric cancer. That's one. The second, besides that, in specifically Hispanic people, there's also a recent study that was published from UT Southwestern that shows that certain mutations that increase the risk of gastric cancer has a higher incidence in patients who are of the Hispanic origin or the Latinx group. So there could be both environmental as well as predisposition combined together might lead to that increased risk of gastric cancer. And we know that the Asian cohort in and of itself has an extremely higher risk for gastric cancer, right? So Asian countries such as China and Korea, they have a very high incidence of gastric cancer, and that's why they have gastric cancer screening programs there. Now, do we see similar rates of gastric cancer in immigrants? Right? First generation, second generation, when does the acculturation kicks in? Those are areas of ongoing research. So there is both environmental factors as well as genetic factors that result in that increased risk.
Thank you. I'm going to go ahead and combine two of these questions and let each of you respond. But first, I want to ask what inspired you to pursue these projects, and then how do you envision your research will change lives? Maybe I'll start with Dr. Halbrook.
Okay, so for me, it's the logical next step in a lot of what we've done in our research. So we've primarily focused on what had been thought as the models centered on looking at the tumor that is the main tumor that we're looking at, I guess. Sorry, I'm not doing a very good job explaining this. So, but a lot of the research, and it's not just our group too, people have primarily focused on this because we've come up with really elegant genetically engineered models to make mice have human disease, and we can study the initial steps into it. And again, we've come up with all these different networks that are really driving how the disease forms and the disease matures. But now, at the end of the day, those aren't the people we end up treating, because if you're eligible for surgery and we can get the primary tumor out, you do great compared to the people who show up with metastatic disease. Those are the people we need to treat. And so it's been a longer time for us to develop good models that we can do, intervention things with gene knockouts and stuff, and look at them in these models that are directly with the metastasis of the liver, which is more pertinent for pancreatic cancer, because a lot of other metastasis models involve the lung, which happens in pancreatic cancer, but it's not the primary site of metastasis. So again, we spent so much time looking at primary disease. For us, the next step was to look at what's going to be the most clinically relevant. And again, I think this is going to be where we will make the biggest progress in terms of treating patients, is studying the relevant disease that patients are showing up that needs to be treated. Because again, if you only have a tumor in your pancreas and it hasn't metastasized elsewhere, excluding some patients where the disease is encroached on the vasculature that can't be operated on, if the tumor can come out, people do far better.
Thank you. Dr. Senthil?
That's a great question. And what inspires us to do what we do, right? To help others. That's it. It comes down to as simple as that. We care about our fellow human beings and want to be able to make a difference in their lives. And that's why I came into medicine. That's why I became a doctor. And then in the field of medicine, right? So surgery was what I was fascinated about. But in surgery, caring for patients with a diagnosis of cancer became my calling. And then once I became a cancer surgeon, what was really important to me is to be care for the most challenging disease process, right? And that is stage 4 cancers, particularly peritoneal carcinomatosis. In peritoneal carcinomatosis, gastric cancer is the most challenging, because the survival for colon cancer carcinomatosis now, with the surgical advances we have, is about 42 months. But for gastric cancer, it's still 10 months, right? When I see young people, 30 years old, 35 years old in my clinic, wanting to be alive to see their kid go to middle school, elementary school, right? I mean, do you need more inspiration than that to be able to help that person in front of you? I just operated on a 34-year-old 3 weeks ago in that stopgap trial. 3 weeks, she has a 2-year-old toddler. What do you do with these people who are in your clinic? If you can't help them, what's the point? What's the point, right? So there's a lot of inspiration. Our desire is to make a difference in these people's lives. And the other question, I did see that. Do we envision that our work will change people's lives? 100%. 100%. I've already witnessed as to what we can do with doing these kind of clinical trials, supported by the basic science work that is being done in Dr. Halbrook's lab, right? So that's the partnership that we have at UCI. Right? We have brilliant scientists like Dr. Chris Hughes, Dr. Halbrook, who are able to provide us with the information that is necessary to translate this into clinical trials, use it in a way that actually will inform treatments that would change lives, right? And that's why it's so exciting to work in a place like UCI. That's why it is exciting to partner with brilliant scientists on the basic science side of things for us as physicians, to eventually make a big impact.
Well, thank you so much. And thank you both very much for all that you do. We do have just a couple more questions. First, for Dr. Halbrook, are you considering utilizing Dr. Senthil's model for analysis of various single or synergistic treatment of specific cancers?
No, we are open to using any model, and that was another advantage of why I chose to come somewhere where, we're bringing a new disease focus into a place that has established other models, other treatment strategies. So really, we have all the tools to look at how our disease would respond to the different treatments that people have available here and how they would behave in the models that are better recapitulating disease that are available to us too. So yeah, I mean, there are at least, again, I've only been here for a few years already, but I have collaborations with people in the engineering department, developing better models. We're working with Chris Hughes's lab as well to see if we can get some of our models into our patient-derived organoid models into his systems as well. So again, yeah, it's again, as mentioned, this is a very collaborative institution where all the investigators are trying to work together with the goal of getting our stuff up to the clinic where Dr. Senthil and others can put them into practice.
Thank you. And then final question for Dr. Senthil, does eating spicy food increase the risk of stomach cancer?
Oh, then I'm in trouble. No, I enjoy spicy food. But anyways, not necessarily spicy food, but certainly smoked food, those things, when there are certain food materials do increase the risk of cancer, but however, smoked foods tend to increase that risk. Now, generally speaking, eating healthier fruits and vegetables, we certainly consider those kind of healthy lifestyle habits generally influence. That is where I think food has a large impact, is what is your general composition of your diet? Is there a healthier version of that diet, right? So more fruits, vegetables, and avoiding red meat for all cancers is beneficial, and it also plays a role in how it changes the internal microbiome of our gut. So that's, if you're going to take something away in terms of diet, so avoiding preserved foods and taking more fresh fruits and vegetables, and that in moderation, everything is okay. Is it true that cancer loves sugar? Cancer, well, I'll answer that, and I'll have Dr. Halbrook answer that as well. And I get asked this a lot in my clinics, like, do I avoid sugar? Should I just avoid sugar altogether completely, right? So cancer is a cell that grows, right? So it's a cell that continues to grow and uses glucose as one of its energy and a fuel. So it is glucose avid. So it pulls in glucose to grow. Now, is eating sugar is how that cancer derives sugar? That's not the case. So cancer gets its glucose whether you eat it or not. It's going to find its glucose, right? But so yes, it does use glucose to fuel its metabolic pathways, but that doesn't mean that completely stopping sugar will also starve the cancer. That is not how the metabolism works. Cancer will get its glucose whether you eat it or not.
Well,
Yeah, and I
thank
see
you.
it's a very hot topic right now in terms of dietary interventions to potentially treat cancer. And there's a lot of data that people do in these preclinical models that involve mice and things like that, which can show strong data for it. But at the end of the day, when we have people show up with late stage diseases, just getting calories into them is a larger challenge than trying to manipulate what calories are going into them. That being said, there's a lot of research, because I work a lot in the metabolic field, extra high fructose corn syrup and things like that. I think it's necessarily how the cancer cells are being fed, but it's how it relates to obesity. I think it's a larger impact. And that is where I think we're seeing such a huge increase in cancers, is through the obesity that's driven through these high sugar diets.
Well, thank you so much to both of you for being here today. And we will be sharing the recording of this presentation as well. So I'd now like to share some information about the Anti-Cancer Challenge with you. We're very excited to announce that the 7th annual UCI Anti-Cancer Challenge will take place on Saturday, October 7th in UCI's Aldrich Park. It will be a hybrid event with opportunities to participate in person and virtually. In-person challenge day will include a 5K and 10K run walk, a 14, 35, 60, and 100-mile road bike routes, a new mountain bike route, entertainment, family-friendly festival awards, and more. Next slide. So we hope that you'll consider starting, joining, or supporting a team, and all proceeds support promising pilot studies and early phase clinical trials like the ones you heard about today. Next slide. And we will be hosting 4 educational webinars in the coming months featuring our renowned faculty speaking on hot topics in health. And for more information, you can visit our website. Next slide. And we're also hosting several free in-person events leading up to Challenge Day, including rallies where you can participate in a training ride, run, or walk, and a research reception where you can tour the cancer research labs on UCI's campus. Next slide. So thanks again for joining us, and please scan the QR code listed here to receive more information. Have a wonderful day.